right, good morning everyone, and welcome to the dissertation defense of Benjamin McKendall, a doctoral student in the Virology and Gene Therapy program. I'm Dr. Autumn Schulze. I'm the current director of the Virology and Gene Therapy program, and it's my really great pleasure to welcome you all here for this important milestone in Luke's career. The defense today is basically a culmination of years of hard work and dedication, and so we're very excited about this, and we're looking forward to an engaging presentation as well as a thoughtful discussion afterward. In a moment, I'm going to welcome Dr. Richard Weil, Luke's mentor, to the podium to actually introduce him. I'm here mostly for logistics, to let you know that this will be followed by a presentation of the thesis work by Luke, followed by a Q&A from the audience. The thesis committee members will then stay afterward for a private defense, so we ask the committee members to hold their questions until that time point, but everyone else feel free to ask as many questions as you possibly want to for this important day. So please welcome Dr. Richard Weil to the podium.
Good morning, welcome everybody. It's a pleasure to have everybody here for Luke's thesis. Luke was setting this up and he said to me before we started, I see you've got about 20 slides. Today's about me, which is not about you. That really epitomizes really what I'm going to, my big problem with Luke. Luke has done an exemplary PhD in the lab. This is just a few of his achievements. He has a review article which is submitted to immunotherapy. He has a first author manuscript which is under review at Nature Communications. He's second or an additional author on multiple other manuscripts. He's been a huge help to the lab in general. And he's presented at external and national and international meetings, and I'll come back to that in a few minutes, or possibly about half an hour. So my view, and I think most PIs here, the view of how a thesis should progress from when the student rotates in the lab and then starts in the lab, chooses the lab, there should be a sort of an increase in the knowledge of the project, reading the literature, becoming completely expert in the literature around it, and then there should be sort of progress with presentations, starting to write reviews, hopefully manuscripts, and then sort of national and international recognition, leading to this sort of independence and that ability to move on and making a student ready to fly. And that's what I tell my students, and I think most people would agree with that sort of progression. But Luke's time in the lab has really made me reconsider and to think, be very careful what you truly wish for. And I'll just give you 4 pieces of data to indicate why I say this. So, on our lab meeting on January the 28th, Jason in the lab put this question: how does the affinity of the Seinfeld peptide for its MHC molecule compare to the affinity of the VSV N52 to 59 peptide? Straightforward question with a very straightforward answer. Now, Olivia Lisseth, who's a very talented MD PhD student in the lab, and who up until this lab meeting I was also delighted to have in the lab, came and said, let's ask Luke. Now, this is my lab meeting. I'm sitting there at the top of the table, pretending that I know everything, and we want to ask Luke. So maybe that was just an isolated incident, but it's really truly not. So there was a meeting, the International Oncolytic Virus Conference in Rotterdam, which I wanted to go to very much. Rotterdam has a lot of ships, and if you know me well, you'll know that I enjoy watching ships. But I couldn't go I said I couldn't go because I'm very important and very busy. It was other reasons. But anyway, the conference organizer sent me a very nice email saying, "Hi Richard, very sorry you couldn't join in person. These things happen, we all understand." That's what I would have liked the email to say, and that's where I would have liked the email to have finished. What it actually says was, but your student Luke did a really great job. I sent Luke instead. He spoke clearly and answered all the questions thoroughly and honestly. So you were not missed. We were hoping to invite him to our lab seminar series in the new year. You see where this is going. Third piece of evidence of why I have to really think very carefully before I get what I want is that we've been very blessed that Hannah Valencia has decided to join the lab. She made that decision in March 2025. one of the major reasons she wanted to come to the lab was to continue this project, which is really the project that Luke will be talking about. Unfortunately, at the beginning of April, I had to spend a couple of weeks away from the lab, and so I sent her an email apologizing, saying, sorry, I won't be there, Hannah, I realize that you'll be completely lost without me, it'll be impossible for you to do anything without me at the helm, but I'll be back in a couple of weeks and I'll try and make up for my absence. She then sent me back an email saying this: hi Richard, please do not worry, Luke has agreed to help me get going, and having him here as part of the project is what really matters. Don't worry about it. And as you all know, science is about egos and huge egos, and particularly don't worry about coming back wounded and deep. So the final piece of evidence just comes from yesterday. Dr. Schultz presented her work. She's a fantastic new PI in the department, and she gave her talk to the DMN, the Department of Molecular Medicine, and at the end she put up her acknowledgement slide, and modesty forbids me to go further, but I'm supposed to be her mentor, helping her develop her career. And on her acknowledgement slides there were all of these people. To be fair, I'm there, but there was a special shout out to Luke Kendall. He was involved, and he was clearly the person that she really was thanking for her success. So, be careful what you wish for. None of this ready-to-fly nonsense. It's clearly a case of goodbye, Luke. It's clear to me at this stage that by far and away the best thing for me, by far and away the best thing for Luke is to move on. To get going. Two big problems. One is he wants to stay in the same field of oncolytic biotherapy, so he will be competing with me. And the other is that he only wants to move up the road to Minneapolis, and I'm not going to say why that is, but I think the relevant people know why. So, there's a major problem for me. I don't want some new hotshot kid, some new kid on the block just up the road competing with me. And so I think what we need to do is to have a complete U-turn in strategy here. And if you can't beat them, you might as well join them. So I decided that the best thing is for Luke to stay here with us, at least in the short term, until I can persuade him to go and work on something like Drosophila eye development, and preferably a long, long way away. He presents no threat. So, I introduce Luke, stealing from the great bard Shakespeare. I come to thank him. He's actually been a fantastic part of the lab. He's set up all sorts of things in the lab that we otherwise didn't have. He's been fantastically valuable to us, as seen by his own additional manuscripts and his own manuscripts. And I'd love to introduce him. These are sort of his specific things. For some reason he likes turtles. He likes tennis players who take drugs. Lauren and I have a very difficult relationship because of what I've just been explaining. And he likes Waldo, which he'll explain. And so it's been a fantastic journey. It's really been great to have you in the lab, and here we go.
Well, thank you, Dr. Vital, for that introduction. I didn't know you prepared that for me, and I don't know how to react to being told to leave the lab.
As far away as possible.
As far away as possible. I'll go to Montana. But thank you everyone who's attending on Zoom, those who came here from work, and those who even came from out of state to watch. I really appreciate everyone's support. It means the world to me, and I hope that at least half of what I say makes sense to you, but we'll see how much we can get. Because, as you can see, the title of my thesis was called Immunodominant Antiviral T-cell Responses Outcompete Immunosubdominant Anti-Tumor Responses to Reduce the Efficacy of Oncolytic Viral Therapy. It seems like Nature Communications liked that title, but it is a little bit wordy. In other words, what I do is I look at oncolytic viruses and I see how they impact the host immune response, hopefully so that we can use both of those to help combat cancer. So my research really is at the crossroads between three major fields of study, which is virology, immunology, as well as cancer biology. So I did my toe in a little bit of each without going into incredible amounts of depth, but because of that, the background is a little bit complicated, but I think it's really interesting to see how all of these fields intertwine and how my project can lay right at the middle of them. So as many of you know, cancer is among the most significant public challenges, the most significant public health challenges globally. In 2023 alone, over 600,000 individuals in the US died from various cancer types, but this is not to mention those who are affected by cancer, the individuals who survived and their family members. It's an enormous issue that's really only second to heart disease. But what makes it very unique is that it's characterized by uncontrolled growth and spread of abnormal cells, which can stem from different cell types within the body, from different tissues, which means that finding a cure for cancer is a little bit of a misnomer, because within cancer, each individual malignancy has an incredibly vast amount of specificity to it. So basically, maybe we'll find a cure for lung cancer or melanoma individually. I would love to see a cure for all cancers, but it's a little bit more complicated than that. Unfortunately, advances in prevention as well as early detection and treatments have improved survival rates for many cancers, and the field of research for developing cancer therapies is really exploding right about now. Historically, what you probably think of when you hear cancer therapies is radiotherapy, which is highly targeted to rupture a solid tumor, or chemotherapy, which aims to deactivate or kill rapidly dividing cells, which are cancer cells. Unfortunately, you have a lot of healthy cells in your body which replicates very quickly, meaning that you are also killing your healthy tissues, and that's why chemotherapy is so detrimental to the patient's health, although it is combating their cancer. So because of that, modern therapies leverage biological agents to have much more specific therapies that don't have as many side effects as you would see with chemotherapy. These include chimeric antigen receptor T-cell therapy, manipulating your own T-cells to target tumors, or in my case, oncolytic viruses, using these agents and their biological mechanisms to lyse tumor cells and ultimately reduce the size of the tumor. More specifically though, with oncolytic viruses, which are the key foundation to my study, oncolytic viruses are naturally occurring or engineered viruses that can replicate in and kill tumor cells. There are many different advantages to using them as opposed to conventional therapies, including natural or programmable tissue specificity, making sure we're only infecting tumor cells instead of healthy cells. They can also be highly lytic, or they rupture the tumor cells, which contributes to the induction of immunogenic cell death, which is the recruitment of other immune cells to the site of infection. Or they can even be armed with immunomodulatory genes, which help improve virus or immune cell mediated oncolysis, or even improve the safety profile of the virus to make sure it's targeting what you want it to target and doesn't replicate out of control. Because just about anyone who hears that you're using viruses for therapy, the first question is going to be, are you going to make the patient sick? Are you going to give them a disease? And that's obviously one of the most important considerations I have when using an oncolytic virus. But luckily, there are many different ways to increase the safety profile. So that's one of the considerations, but the other is when you're using a virus, you also need to make sure it's not shut down by the host immune response. So mechanisms like viral genome sequestration and neutralizing antibodies can neutralize your virus. So you also need to consider how it can be efficacious in the face of antiviral immunity. So what you're going to be hearing me say dozens and dozens of times in this presentation is CD8 T cell, or T cell specificity. CD8 T cells are cytotoxic T cells that are the main effectors for killing an infected cell in the case of a virus infection. So we want to target those to see if we can also use them to kill tumor cells. They develop in a very dynamic process that begins with what's called antigen priming. So viral proteins or foreign antigens are taken up by antigen-presenting cells and then presented to these naive resting T cells in priming. And then that leads to their activation, their expansion, so you get a large population that then migrate to the site of infection, ultimately killing your target cells. It's a very dynamic process that peaks and then contracts, leading to immune memory, which is what we see with vaccines, and that's why we use vaccines, it's for immunological memory. But the key here is antigen-specific immunity, and that's not just about the viruses, because in theory, you should be able to prime an anti-tumor response against tumor antigens as well, the proteins that are expressed on tumors. So in a perfect world, you use an oncolytic virus, you generate this antiviral response that we would expect, but also we generate this sort of immunity to cancer, is the very simple way to put it. Unfortunately, putting it simply is not going to be very beneficial when it's an incredibly complex dynamic. This is why Dr. Weil mentioned Where's Waldo. It's kind of what this project's been known as for the last 4.5 years, is the Waldo study. And that's because there's a term called immunodominance that we explore, where the antiviral response dominates over the anti-tumor response. Your body spends so many resources reacting to the virus that it's distracted from the tumor itself. So basically, you infect with an oncolytic virus, you then get a circus of viral antigens, thus obstructing your Waldo, your tumor T cell that wants to bind its tumor antigen. As I said, this has been known as the Waldo Project. In fact, we refer to the grant it's on as Waldo Grant. I get emails from other PIs anytime they see the word Waldo. I get some very scary emails from Dr. Mary about the death of Waldo. So this is all to say that I'm going to run this metaphor to the ground, and we're going to see if it ever needs to be used again after that. So leading into that, what we're trying to do is find tumor antigens in the circus of the antiviral immune response. So we're trying to optimize this interaction between a T cell and its tumor antigen. But you introduce an oncolytic virus, you get a high concentration of viral peptides, as well as the expansion of antiviral T cells, possibly at the expense of the T cell interacting with its tumor antigen. It gets much more complicated though when you consider the nature of central tolerance. Because during T cell development in the thymus, T cells which recognize your own antigens are selected against and undergo apoptosis, basically as a means of preventing autoimmunity, because you don't want your own immune cells recognizing your own proteins. The problem with this though is that tumor proteins are your own proteins, which means that the T cells that we have to work with are already being killed by your own immune system as a means of self-preservation. So that means that the only T cells we have to work with are ones which have escaped this selection mechanism, which makes them inherently very weak, even if they do bind their Waldo antigen. But even presuming that you have your tumor antigens, you have your tumor T cells, it's been shown by data from the biolab, beginning with Dr. Laura Evgen, showing that your anti-tumor T cell can actually undergo attrition or exhaustion in the face of antiviral immunity. So if you have a very immunogenic virus, those existing T cells can actually become shut down. So what this project does is it challenges the paradigm of oncolytic virotherapy, which is generally considered that you use an oncolytic virus, you prime antiviral immunity, and you also get some level of anti-tumor immunity. We are saying that that's not necessarily always the case. This is optimal oncolytic virotherapy, where you have your antiviral and anti-tumor T cells being generated at equal rates, being able to kill the tumor even if it's not infected. We are proposing that with the essence of viral antigen concentration, the nature of central tolerance, both of those mechanisms contribute to making it more difficult for the anti-tumor response to thrive. So with all of this background and all these considerations, we also need to focus on what makes a CD8 T cell response beneficial for our therapy. For those of you less familiar, we need to acknowledge the specificity of the T cell. We need to acknowledge the abundance of the T cell in the microenvironment, since they're the effectors that are killing the cells. And finally, their functionality. They can be in the tumor, but they can also be senescent, which means that they're not doing much for you. So it's a lot to process and a lot to consider when designing a T cell or T cell based therapy. But all of this leads to our central hypothesis, which is that an immune response that's stimulated by highly immunogenic oncolytic virus can immunologically distract from the priming, proliferation, and cytotoxic activity of anti-tumor CD8 T cells. So the overarching aims of this research is first to identify and characterize this antiviral immunodominance. I've been speaking of it so far as kind of a given that this is what's seen in the field, but there actually hasn't been substantial data to support this claim, at least directly. So my study is to actually provide data showing antiviral immunodominance over antitumoral. But identifying a problem isn't very good when you're just trying to actually develop therapies in the long term. So we tried to figure out how to overcome antiviral immunodominance to promote therapy, because we come from an angle where we still want to use oncolytic viruses. This isn't necessarily against them, because there's many different benefits of using this as a therapy. We just want to find a synergy with the anti-tumor T cell response. We explore these aims using vesicular stomatitis virus as our model inflammatory oncolytic virus. It's within the family of Rhabdoviridae and is a negative single-strand RNA virus with a primary tropism in cattle, horses, and pigs. What this means is it doesn't replicate particularly well in humans, which is great as a cancer therapeutic because it means the virus can only thrive in malformed cells or dysfunctional cells like cancer cells, which means if it infects a healthy human cell, it can generally be cleared. But it's not just a model for, or a toy for me to study, it's actually has a very healthy presence in preclinical and clinical trials, where a construct has been used for treatment of multiple myeloma, non-Hodgkin's lymphoma, again in the clinical setting or clinical trial setting. And there's also a construct being tested that expresses tumor antigens for metastatic uveal melanoma. So it's a very robust oncolytic virus, and it very much serves the purpose of stimulating strong antiviral immunity for the purpose of our study. So the first aim that we explore is to determine the relationship of antigen concentration and establishing the balance of antiviral and anti-tumor immunity. The simple fact is, if there's more tumor antigens, does that make more anti-tumor T cells? Now, we do this in a couple different ways. Again, this is within the context of the Waldo circus, where we can model the antiviral response without using viruses. We think a lot of the viral proteins are what's obstructing the anti-tumor response from thriving. So we can use immune adjuvants that stimulate immune pathways that ultimately lead to an antiviral response. This is triggering genome sensing pathways from various pathogens, but without actually introducing those pathogens themselves. In other words, we are eliminating the circus and still being able to find our Waldo. At least that's what we hope. On top of using these agents as part of our experimental groups, we also use we always use subcutaneous B16 melanoma as our tumor model, and it expresses ovalbumin. Throughout this presentation, you'll be seeing ova and ovalbumin. Any T cell that recognizes these antigens are what we consider an anti-tumor T cell. It's our model, and it's and how we balance antiviral and anti-tumor T cells is through the lens of ovalbumin. Or in other words, synfecal, which is part of ovalbumin. So these two are interchangeable. But finally, we have a VSV construct which encodes for ovalbumin, our tumor antigen. In other words, we are generating more tumor antigens, so then more Waldos make it easier for the T cell to find. So we've designed a couple different scenarios, and we can finally implement that into a mouse model, where we use immunocompetent C57 mice. We inject our B16 ova tumors, and then we administer our treatments intratumorally. Around day 7 in mice within this model is when we would expect peak expansion of antiviral T cells. So at this point, we harvest spleens and tumors, and we can actually see the T cell populations that penetrate the tumor, and we can determine if they are specific to virus or tumor. We also administer OT1 T cells. These are T cells which have transgenic T cell receptors recognizing ovalbumin, meaning that we're artificially implanting a large population of tumor-specific T cells right at the beginning, which makes it as easy as possible for our therapies to expand an anti-tumor response. And this becomes relevant as we move forward, but this is an artificial system where we are adding in anti-tumor T cells. So we would hypothesize that minimal expansion of these anti-tumor T cells are observed in the presence of your inflammatory virus. I also included this. We're mostly looking at the phenotype of the T cells, but within this model, using our VSV expressing tumor antigens plus OT1 is in fact curative. But now the question is, why is it curing mice? And just to orient you on a little bit of flow cytometry, you'll be seeing a lot of these figures, so I wanted to show you how to interpret them. This is looking at the T cells within our tumor, and on the vertical axis you have tumor-specific T cells, on the horizontal you have virus-specific. So the agent that we used for this generated both tumor and viral-specific T cells. I show you this because it's a whole lot to look at over here, where across the spleen and the tumor, across 4 separate mice, we can see some common trends. So these are our non-viral immune adjuvants which stimulate the immune response, but we didn't see an expansion of anti-tumor T cells in either the spleen or the tumor. What we did see, when we did see expansion of anti-tumor T cells, is when our virus expressed the tumor antigen, or our Waldo, with or without that artificial population of T cells that we introduced. So this is a very potent way to establish anti-tumor immunity. But what was actually most striking to us was that VSV on its own, GFP is just a control, was not able to expand these OT1 T cells. We would have expected at least modest expansion of OVA-specific T cells, but because we did not see any, that's actually alerted us to the whole nature of immunodominance, because we would expect at least a marginal expansion of these T cells because of the way the virus interacts with the immune system. We also validated this by taking those T cells and pulsing them with our the relevant peptides, where we can see a synfecal or OVA-specific response is found only really in the VSV OVA T cell groups. But that's all to say that we have found two scenarios with this, that either VSV replication and T cell killing doesn't generate enough tumor antigens to prime those OT1 T cells, or the antiviral response can lead to the death or attrition of those T cells, or it could be both, but at this point we hadn't characterized it. But what we also did was take this outside of the realm of B16 melanoma. We also reproduced this in colon adenocarcinoma cells, just to verify that what we're seeing is not just tumor specific. And we found in the MC38 expressing our tumor antigen that there's not a significant expansion of OVA T cells with VSV like we see with VSV OVA, showing that VSV inherently is not going to generate an extremely potent anti-tumor response like we would have hoped. So overall, with the first part of this study, we see that non-viral immune adjuvants that I mentioned before, as well as oncolytic VSV on its own, are unable to expand these high-affinity anti-tumor OT1 T cells in vivo, even though we're kind of giving them the best chance that they have. So the next question from this is actually looking at the types of T cells that enter the tumor, not just specificity, but functionality. By doing this, we are able to identify T cell populations which correlate with improved therapy in the mice. So we reproduced a version of this study where we looked at mouse survival, and this is in the absence of OT1 T cells. This is purely an endogenous response. When you treat with VSV and tumor antigen, you see a shift in the survival curve to the right, improving mouse survival. We also see the generation of tumor-specific T cells, as well as tumor-reactive T cells, in both the spleen and the tumors, just showing that we've reproduced our findings that I showed you in the last paper. But what we're actually taking away from this is looking at the T cell subsets that are within the tumor. And what we found was that although VSV OVA, our tumor antigen expressing virus, leads to improved survival, the phenotype of the T cells are very similar between VSV GFP and that survival construct. I put little brackets because there's a lot to look at here, but the real conclusion is that between VSVs, the populations are extremely similar, both in CD3, memory, CD4 T cells, even across T cells which are exhausted, have different functionalities in the tumor microenvironment, or express different markers. They're very similar between the two groups. So our question is, other than specificity, why are these T cells promoting the survival of the mice? And we're actually able to explore this with more depth when we use mass cytometry by time of flight. It allows us to look at a large panel of markers on each individual T cell and cluster them based on their overall phenotype. So we were able to actually identify T cells that are terminally exhausted, those that are nearing exhaustion, those who express certain markers. I won't go all the way down the list for the sake of time, but basically we're able to put them in little boxes and then see which ones are expressed more with our VSV OVA treatment group. And this is actually a good visual representation of the antiviral circuits that we're talking about, where these clusters represent certain types of T cells, and these are our non-viral treatment groups. What you notice is that when you go from here to our virus-infected groups, the clusters shift dramatically to the left, which suggests that the VSV is dominating the immune response. These are in response to the virus. We also see these in our VSV OVA treatment groups. And the fact that these exist here and they don't exist here when there's no virus shows that there is this dynamic shift in T cell phenotype population that's modeling kind of that the circuits that we were talking about before. But what's interesting is that it's hard to see here, but I'll show you in just a second. VSV OVA actually rescues some of these populations that we saw in the absence of virus, showing that the type of antigen you use can affect the T cell population overall that's infiltrating the tumor. And this is what I was alluding to. Again, for the sake of time, I won't go into depth about these subtypes because even more research is required for them. But for a correlative study, we see that between VSV and VSV OVA, there's an increase in these CD39 positive T cells, these moderate exhausted T cells, as well as those exhibiting short-term effector function. So this leads down a longer path of phenotyping the T cells, seeing their cytotoxicity, but at this time we're able to see that there is a dominant effect of the antiviral response, and that we can actually see unique subsets of T cells generated when we have tumor antigens. So the takeaway from this is that VSV induces that shift in the T cell landscape, and thus contributes to our observations of immunodominance. So to summarize AIM-1, I have the general summary on the right, but for more descriptive, we see that VSV alone in this model doesn't generate that optimal environment that we want for priming anti-tumor T cells. but also those clonal populations of T cells that we generate seem to favor an antiviral phenotype, meaning that, again, this dominance is observable when we look at phenotypic properties of T cells. So we've shown you the circus, that's what we're doing to identify the problem, but how do we actually overcome this in order to promote therapy? So since this is very T cell focused, it makes sense to combine oncolytic virotherapy with other immunotherapies that also target T cells. And the first one we look at is immune checkpoint blockade. So immune checkpoint blockade therapy is a therapeutic approach which aims to shield T cells from the hostile tumor microenvironment. Each T cell, as a regular biological mechanism, expresses regulatory proteins, or otherwise known as checkpoints, which means if those proteins bind their reciprocal ligand, they deactivate the T cell. This is so your immune system doesn't go out of control. The problem with this is that many tumors have mutated in such a way that they take advantage of this checkpoint. And even though a T cell comes in to kill a tumor cell, it expresses the thing that activates the checkpoint and ends up killing the cell before it can kill the tumor. What immune checkpoint blockade therapy does is it shields this interaction from occurring so the T cell can still kill the tumor. So basically, it's making our anti-Waldo T cells stronger. And we rationalize this based on our previous aim of showing immunodominance, that early expansion of T cells will largely favor the antiviral T cell population due to many different things, including that of high antigen concentration early on in viral infection. Which means if we treat early with immune checkpoint blockade, we're shielding virus-specific T cells. If we treat later, we are salvaging the anti-tumor T cell response that is generally lagging behind. So we explore this early and late administration of our combination therapy in order to see if we can promote anti-tumor T cell expansion. We're introducing a new concept here, which is that of antigen affinity, where we're testing VSV, the VSV OVA, which is again a non-self high-affinity tumor antigen, as well as one expressing GP100, which is an endogenous melanoma antigen binding with very low affinity to its cognate T cell receptor. And this has implications for the antiviral response that I'll hopefully be able to explain within later slides. Essentially, what we found was when we combine these therapies together, we see that VSV OVA with early administration of our immune checkpoint blockade, which is targeting programmed death protein 1, or anti-PD1, we see improved survival in the mice, showing that the antiviral and anti-tumor T cells are benefiting from therapy. The real test here was to see if a low affinity endogenous antigen could also benefit from this therapy if we delay treatment. But regardless of early or late administration of PD1, we did not see an improvement of survival in the VSV GP100 mice, suggesting that in order to have successful combination therapy, you need to consider the antigen affinity as well. Only if we have our very fake artificial antigen can we see improved therapy. But in the real setting, we really don't see too much synergy. So we found that combination of these two eliminates tumor only when potent tumor-associated or tumor-specific antigen is expressed by the virus. But now this poses the question of affinity. What kind of Waldo are we looking for? Which ones will actually be able to generate a tumor-specific T cell response? So that gets into the final part of the presentation, as well as the end of AIM 2, is to examine the influence of antigen affinity on the establishment of tumor-specific T cells in the context of oncolytic virotherapy. Now, I know this has been kind of complicated up until this point, but don't worry, it's going to get much worse. So we're going back to the nature of central tolerance when we're developing T cells that can recognize tumor antigens. The problem with this, again, is that antigen affinity, which our tumor T cells to work with, bind with very low affinity to tumor antigens already. This has implications for the potency of your therapy and the potency of the expansion of the T cells. So what this means is that our current model that uses ovalbumin is not necessarily sufficient because it's very high affinity, it's very fake, it's not actually showing us what it looks like to expand against a low tumor antigen or a tumor antigen with very low affinity. And because central tolerance does delete those potent self-reactive cells, it restricts our options for generating tumor-specific T cells. So we need to design a model that actually lets us study this in the context of oncolytic virotherapy. In other words, we're trying to figure out how to make Waldo easier to see. We're not affecting the circus, we're not adding more Waldos necessarily, but we are actually trying to make Waldo easier to spot. And we can do this by mutating that tumor antigen in such a way that it's no longer a self-antigen that's tolerated by the immune response. And further, in the model that we're going to design, we want to look at varying affinities, because there's going to be a threshold where we have potent affinity and you have low affinity, and there's a whole spectrum within it that we want to be able to look at individual points in the affinity spectrum. So we're able to do this. First, this is not in the context of tumors. This is just setting up the model where we use our ovalbumin and OT1 T cell model to establish that foundation for a functional affinity gradient. I say functional affinity because the data that we use in order to design our altered ligands here is based on OT1 sensitivity to the individual peptide. So this is 1-to-1, very strong, and then as this goes down the line, this is almost a third as potent, going all the way down to 18-fold decrease in affinity. So you go down the step ladder as you mutate single amino acids within this individual epitope. In other words, we have our high affinity binding synfecal and our low binding sikfecal, which is the Q4 variant. So we have this model from a previous study produced this model, but what we did was take this and put it into VSB, our oncolytic vector. And what we're able to do by inserting these proteins is that now we have VSB expressing high affinity protein as well as VSB expressing low, and also a couple in the middle, which is very valuable. We validated this in vivo, where we wanted to expand OT1 T cells in a mouse to make sure that these proteins behave as we would expect in relation to the T cell response. So we put in OT1 T cells and we have our virus, which is expressing those tumor antigens, and we actually see a decrease in the expansion of anti-OVA T cells as you decrease the affinity gradient, which is what we would have expected, but sometimes in mice things behave weirdly. So it's very good to see that as you decrease the affinity gradient, you see a corresponding decrease in sensitivity to synfecal. So that's all to set up the model and to design constructs. But where we actually bring this into the world of therapy, into the world of tumor therapy, I should say, is in a mouse model that endogenously expresses OVA. And that's in what's called a RIP-OVA mouse model, where it has strong expression. Oh, sorry about that.
It's the right more, yeah. Wait.
Okay. I'll take this. All right, so we're taking the model that we set up and we're bringing it into a RIP-OVA mouse model, which expresses membrane-bound ovalbumin in pancreatic beta cells. And what this means is that while we've been using this artificial OVA, right now in this mouse, it's tolerant to ovalbumin, so meaning it's a self-low affinity antigen. Again, expressed on pancreatic beta cells. So basically this affinity gradient has swapped, where low affinity is going to be that endogenous antigen. And as we go all the way to that Q4 variant, which is very different, I can't say low or high affinity now. Right now it's less self. That makes it bind with much higher affinity. So now we've kind of flipped the script on this model where we can look at low, medium, and high affinity antigens in the RIP-OVA mouse model. One thing that's very relevant for this model is because ovalbumin is expressed on pancreatic beta cells, if we make anti-tumor or anti-self T cells, that means we are killing cells in the pancreas, which induces a diabetes phenotype. So what we're able to do is detect blood glucose levels of mice that were fasted for 4 hours after treatment, and see that when we used our high affinity Q4 variant, we were able to, indirectly, at least this is how we're interpreting it, kill pancreatic beta cells, leading to diabetes in the mice. So we are inducing autoimmunity, which is generally a pretty scary thing, but in the context of tumor therapy, that's actually what we're searching for. So our constructs in this mouse model can induce some level of autoimmunity. One thing to consider though when looking at the affinity spectrum is to mutate the tumor in such a way that it's easier to see, but you don't want to mutate it so far away that it becomes something entirely different, because then our anti-tumor T cell won't even recognize anything on the tumor. So it's a very delicate balance as to where you mutate the T cell, or sorry, the antigen, so then you still get therapy. So we found that VSB expressed near cell tumor antigens can expand autoreactive CD8 T cells, and it's detectable by flow cytometry. So this is where it gets very interesting, and unfortunately this is one of the last studies I did in my PhD because there's so much more to do from here. But when we look at our variants here, this is VSB ovalbumin wild type going down the line to our high affinity, we see two different populations of T cells that are generated that can react to a tumor antigen. The first being the Y3 variant, which generates T cells against N4. This is purely a tumor antigen specific T cell population. meaning we have broken central tolerance by using this variant. And we don't see it nearly to the extent with our near self antigen here, which is laying just about threefold lower than the wild type, or threefold higher depending on how you look at it. But even more interestingly, the other population we see is one that is specific to our new antigen, our Q4 variant here, but those that can actually cross-react with that N4 T cell. So we've used our new antigen to generate T cells specific to that one, but the T cell receptors are formed in such a way, or the clonal populations exist, where it can attack tumor cells. It can attack the tolerated self-antigen. So these are two entirely different approaches in order to break central tolerance, where we use the ones that will likely make you very sick if there's a lot of them because it's very self-reactive, or we can use a smaller population of cross-reactive T cells. It's kind of a lot to process and a lot to interpret, and we don't have a ton of answers right now, but it was very interesting to see that the antigen affinity gradient can generate unique populations of T cells that may have implications for therapy. For those interested, these are some quantitative results. It tells the same exact story, it's just the same data, that we see a large population of N4 reactive T cells with Y3, and this is also recapitulated in the spleen. So the point of all this is to look at the threshold where anti-OVA T cells can be generated, and that seems to be around 4-fold non-self. And then if you go all the way down to nearly 20-fold less, then you can create new T cells which recognize your tumor antigens. Moreover, when we look at these T cell populations based on physiology, phenotype, exhaustion, we can see a difference between them, especially when you look at markers of activation and exhaustion. In short, the bright red here, which is PD-1 positive, LAG3 negative, these are functionally active T cells generally, whereas the dark red are exhausted. And you can see across the dual reactive, which is right here, and the single reactive ones here against the Q4 or N4, there's different physiologies of these T cells overall, which means that we're not just generating T cells that have different specificities, they actually behave differently in the tumor. And this might be because they've entered the tumor earlier or any number of other biological factors, but we're actually making T cells that are different in physiology, which is fascinating. And in addition to this, when we look at the TCR, the clonality of the T cells, the variability of the T cell populations you're making based on their T cell receptors, between the Y3 and the Q4 variant, you don't need to know what each of these individual ones mean. What we can see is that there's a difference between the two, that we're actually generating entirely different populations of T cells, which can recognize different antigens. Again, a lot of characterization is needed for this portion, but the distinction between the two groups is the key takeaway here. So in other words, when you are mutating your antigen, we were able to find a threshold or a sweet spot where you can generate anti-tumor immunity without going so far that you can no longer recognize the tumor antigens in the first place. So we can kind of play around in this space to see which of these T cells are the most therapeutic. As of right now, with the experiment that we performed, we didn't see a significant increase in survival when we used our less self variant, the Q4. It's not significant here, but you can see there was extended survival. But what is visually observable is a delay in tumor growth in the presence of these dual reactive T cells that we made, which means that they may play a role in therapy if we somehow find a way to boost these T cells. This is all single dose administration of our vector, so if you were to boost potentially around here with the Q4 peptide, you might be able to salvage the T cells present at this point in time, which might promote survival. This is just one of many experiments that we can do in order to see if these are actually therapeutic, but it's still something that's worth pursuing, even though we don't see a very nice survival curve at this time. So overall, with this portion, we see that virally encoded near self tumor antigens, I put in heteroclitic here because that's a term that we use to describe dual reactive. They resolve the conflict between that dominant antiviral and subdominant anti-tumor T cell response, overcoming that antiviral circuits that we were very worried about. Again, I have my general summary on the right, but to be more descriptive of part 2, our anti-PD-1 immune checkpoint blockade therapy is most potent when we have that high affinity tumor-associated antigen. In fact, it's not even effective at all in our model when we use the low affinity GP100. And then also with the B16 OVA model, there's that threshold of near self or far selfness that actually matters for the generation of anti-tumor T cells and breaking central tolerance. It's unclear if this is VSV dependent, but the fact that it can happen in the presence of antiviral immunity is very promising for our approach to therapy. But there are also multiple models that we can explore this whole affinity gradient in that we would hope to do in the future. So the key findings overall in conclusion are that in the context of highly inflammatory oncolytic virus infection, you can see the ablation of low affinity anti-tumor T cell populations, or at the very least, a lack of expansion of those T cells, modeling that version of immunodominance, which also extends to the phenotype of these T cells, their functionality. But then if you combine them in a certain way with the other immunotherapies, you can see synergy if your tumor antigen is of the right quality. However, if that's not an option to you, you can actually mutate mutate that antigen in such a way that you can activate endogenous T cells against the tumor. But it's a very refined process and hopefully going to be generalizable someday, but it's very specific to this model at this point. The next steps, of course, would be to look at those dual reactive T cells, see their cytotoxicity in vitro. It would also be good to explore those ovalbumin mutants in combination with immune checkpoint blockade. So basically combining both parts of AIM2 to see if there's benefit in therapy. And then also just further looking at the TCR clonality between these T cell populations, as well as more creative vaccine boost strategies in order to promote therapy. Like I said, there's a million open doors with this project, which makes it very exciting, and there's still a lot more to do. But with that, I'd like to get into the acknowledgements. I'll try not to take too long, but it's still very important. I'll start with thanking Mayo Clinic and the graduate school as well for academic and financial support, especially the NIH T32 Research Training Grant, which funds the students in the department, and the Department of Molecular Medicine for their contributions, and of course, Dr. Richard Vile for paying for my PhD out of his pocket. I'd like to thank also my thesis advisory committee, which is comprised of experts in the field who have followed my progress at least every 6 months, but I interact with them very frequently during my PhD, and they're instrumental in the designing and the execution of these experiments. But on top of all this, I'd like to thank my lab. Everyone is phenomenal in that lab. People come and go over the years, and I've never been in a point where there was any tension in the lab. There wasn't like a weird social thing that you can see in some workspaces. I really like how the people are, and obviously on top of that, I like how much they helped me, because they certainly did. Special shout out to Jill Thompson, Tonic, as well as Olivia, who directly worked on these experiments with me as well. And I'd like to thank Dr. Richard Vile, who is an incredibly hard worker, incredibly jokey, very funny guy, but his compassion for his students and his lab, and maybe most importantly, the people in the clinic that we're actually designing therapies for, is absolutely overwhelming. And I'm amazed that he can do this day in and day out. And with all this, it very much makes up for his insufficiencies in axe throwing. He can't be good at everything, and that's okay. But what's most important these last 5 years is that he's done an expert job at balancing the different roles of being a PI, where you have to be a boss, you have to be a mentor, and you have to be a friend. And this is something that changes every single day in order to make sure that your student is as proficient and as capable as they can be. And some days, you have to be more than a boss than a friend, and also since I just showed data, it's always good to show some statistical stuff. So thank you, Richard, for everything you've done. So when I was practicing the acknowledgements, I originally put Lauren at the end, but I couldn't make it through it unless I had momentum from the presentation, so she got bumped up quite a bit. Lauren is my fiance that I met in 2020, like 2 weeks after starting graduate school, and in 2 months after graduating, we're getting married. So I think that's a great way to bookend the PhD experience, and I can't think of a better way to do that. But she's been absolutely incredible, and I really don't know how I could survive long nights in the lab and the regular struggles that adults have without someone by your side. So I love you.
Me too.
I also did want to mention that I'm graduating a little earlier than most. Usually it's like about 5 years, but I'm shooting for 4.5, explicitly so that at our wedding there can be 2 doctors. I can't have one. I'd also like to thank my family. Oh, this one was also hard to get to. So my brother Jacob and my sister Rebecca have been with me my entire life because I'm the youngest, so I literally don't know what life is like without them. I put the Waldo hat in case you couldn't tell, but this is obviously. And they've done everything to make sure that my life's a good one. And I feel like it's not a given nowadays to stay close to your siblings when you grow up. Life's hard, but I feel really grateful to be able to say that they're still my incredibly close friends, and we don't really fight too much anymore, and I think that's really good. And I'd like to thank my parents as well, who flew out to support me. One thing I told myself I'd get through before I just completely choked off is that when I was in undergrad, I was planning to go to med school, and obviously that didn't happen. So there was a couple of years where I didn't really know where I was going, but my parents were always absolutely with me every step. So thanks for that. See, if I were to do Lauren right now, I'd be done. Okay, I'd like to thank Lauren's family as well. Moving here and then being close with them nearly right at the beginning, I've always had a family away from home. They also made sure that I had at least one meal a week that wasn't frozen or from a package, so I really appreciate it. I felt like part of the family from day one. You're all absolutely incredible. I'd like to thank my different subsets of friends. First is the ones I met since graduate school. I'm not very good at taking pictures, just know that there's more than just these people, but these are the ones that I had on hand. It's amazing to have so many contemporaries in the field who are interested in the things that you are as well, especially when they're brilliant scientists, because when they become famous, I'm gonna have a lot of coattails that I can ride on, so each and every one of you, I'm counting on you, so please don't let me down. And then all my friends back home, some of which are able to attend on Zoom today. I don't have time to go through each individual person, but everyone has unique qualities, unique strengths of character that I truly admire, and I use that to make myself a more well-rounded person. So as an adult, I have my friends to thank for who I am today as well. And with that, I truly thank everyone for your time and for listening to all of my research. Obviously, I've done more, so I'm happy to talk. I'm happy to discuss anything that you may want to talk about, and I'm not sure if everyone was able to find Waldo. I kind of threw this picture up a bunch of times, and you had some time to find him, but Waldo's not actually up there, even though I circled it, because he's right here.
So, something that Dr. Schultze did not explain is this is the first year in the EGT track that the PhD exam has an extra component, which is called the presentation clarity score. And so what we did before we started here was we emailed very random people in the audience, completely random set of people, to find out, to get feedback on how clear Luke's research has been. And so we're just going to present that now, take a few minutes, so and then we can have questions and answer session after that.
Howdy, congratulations.
Hey Luke, congratulations.
Hey buddy, it's me. Dixie's here as well.
Hi, this is Aunt Marcine.
Hi Luke. Hi Luke.
Congratulations, Luke.
Oh, hi there.
Hey Luke, it's Aunt Meryl.
Hi Luke. This is Rebecca, your cousin.
Hello, Luke.
Hello there. Hi Luke. Nope.
Hey Luke, I've gathered all of your closest family and friends here today to first wish you congratulations on all the hard work you've done over the last few years. I know there's been a lot of people who are really important in your journey that unfortunately couldn't make it here today, and so we wanted to include them virtually. And second, more importantly, we wanted to promote how smart you are by the best way we know how, which is showing how dumb we are. So I tasked all of your family and friends to describe what your research is and what you do at work each day, and the results are pretty good. So without further ado.
That's cool.
I just tell people you do oncolytic virus research or like cancer research, because most of the time people don't know what I'm talking about. And that's the gist of what I could say. I know it has to do with T cells.
Luke does is he's going to take viruses that are already in the human body and figure out a way to attach them to various and sundry cells to cure the cancer, which I think is really awesome.
Luke's thesis is on
oncolytic virotherapy. I want to say that's how you pronounce it. I could be wrong. Also known as the premise for the movie I Am Legend. So, one sole goal for his life is to be the only man left living on the planet. And fight the occasional zombie that he created. So, he's going to give it this fancy term and talk about spitting viruses and these viruses that cure cancer and all that kind of stuff. But ultimately, this is all for his own personal gain. So he can have rule of the entire planet and he can do it what he wants.
Also is cancer, but then there's a tick. So I believe that is just to separate the word, okay? So he's studying the tick virus and teaching them how to find cancer cell and eat them so the cancer patient won't have to go through chemotherapy.
You think you play with mice?
All day long.
Yep. And we try to solve cancer with genes from the mice.
Viral
Oh, that too.
He uses viruses.
Virus to kill cancer.
Yes, that's what he
Maybe.
does. Yes. And I think he's looking for Waldo.
In order to solve the cancer, he experiments at the Mayo Clinic, where he researches every day.
And I'll say Waldo's the first name that appears, which makes Waldo the first author on Luke's own thesis. I mean, that's the only critique I've got, but simple enough. I think I've got that down. Oh, there's a second part. Finding tumor antigens in the circus of the tumor microenvironment. Oh boy, okay. All right. Finding tumor antigens, antigens. What are gens? In the circus of the tumor microenvironment. That circus does not sound fun. I do not want to ride those rides.
I'm sure you've heard of me talking about how he uses a horse virus to fight the cancer. So what that means is there's a virus, or no, there's a horse virus. So they take a horse, bring it out.
He then goes to his desk to grab the cancer off the shelf, and any animal, preferably a mouse, he can get his hands on, to grow tumors. Once the tumors grow, he looks at it with a microscope. He then writes down on his clipboard what it does. That's a secret. He's not telling you or me. And then he puts it on a big poster board and goes around the country showing the flow chart of said tumor.
I know you're going to cure cancer, Luke. That's all that I know that you study is oncology.
Research?
No, something with viruses.
Viruses.
Virus oncology.
Virus oncology.
Viral oncology.
Viral oncology.
I don't know.
I think of you as Mario. And I think of you out there and you're trying to go get Princess Peach. And Princess Peach is the cancer. And you are chasing it, you're going over the hills and you're going down in the valleys and you're doing everything that you possibly can. Because your job is to cure cancer. I know that. I can't remember all the big words, but I know you're going to cure cancer.
These guys are wise. That's
I
cool.
realized it was for research. I thought that you were just killing mice for fun. The whole for a purpose component is news to me.
I was thinking that you were going to be some sort of an assassin. That's where I was leaning with, that you're going to go international assassin. Then they told me it had something to do with cancer research. I didn't know that that was true or not. I still think it's a cover story.
And I'm not quite sure what you do, but I know you take a little Reese's and maybe cut them open.
Mostly kills mice. But he's gonna say something.
Another term, like the true crime term of Black Widow. It's like when the wife slowly poisons her husband to death, but like nurses him back, like tries to nurse him back to health. That's what I think Luke does. He gives his poor mouse wives cancer and nurses them back to health until he slowly sends them off to the wheel in the sky.
Yeah.
But all for a good purpose.
Sure. Presumably. But yeah.
Ignore that. Luke, I'd like to congratulations. I'm really, really super proud of everything you've accomplished.
I'm very, very proud of Luke and all the work that he's done and all of the countless hours and years of his life that he has dedicated to this program and this career that he is stepping into. And yeah, I just wish him all the success. I know he's going to kill it and keep being the super nerd that I know him to be.
You put the time in, you deserve being the doctor you are. So good luck for your future.
I know you're gonna cure cancer, and you're going to do it because you're one of the smartest people I've ever known. I love you, buddy. Congratulations, Luke. Keep at it, buddy. See you soon.
You are objectively one of the smartest people that I know, and I know that you're gonna do amazing things, both after your PhD and just in life in general, and I think you've been an amazing friend in my adulthood, and then also one of my great colleagues, you know, when we were both at the NIH, and so I appreciate you, I'm proud of you, my mom sends her congratulations, and I think you're gonna do amazing things, and I can't wait to be in your corner every step of the way.
You're gonna be good and awesome, and so, we're so proud of you, we're so proud of you, you're one of the smartest young men we know.
We're super proud of you. Can't wait to see what your future has in store for you.
Thank goodness we have people like Luke to defend this and explain it to us normies. Congrats, Dr. Kendall.
You really have such a huge passion for the work you do, and it really inspires those around you to be better and do better. I'm just really excited to see what you do in the future, and really hoping someday that I can say my husband cured cancer, so no pressure.
For these last 12 years, it's amazing to see all the progress that you've made and the impact that you're going to have on science and medicine. We wish you all the best.
We love you, Lou.
That's
It's
cool.
not always gonna be successful. You're not always gonna cure cancer, but it's about the tumors you made along the way.